So in the last video, we talked a lot about the structure of membranes. So we looked at the kind of composition of phospholipids as well as proteins that were attached to it. So we know that membranes are semi-permeable and will only allow certain things to get through. So we're going to be talking more about how things can get through membranes when they can't always diffuse. So there are a few ways for a molecule to get in or out of a membrane. So the first category is passive transport. So this just basically means we're not going to expend any energy in the form of ATP. So we can use other tricks to help bring molecules into the cell, but we're not going to use ATP, and that's the trick to passive transport. So one way that molecules can get into the cell and out of the cell is by simple diffusion. So on the next few slides, we'll be looking at the sort of characteristics that allow a molecule to go in and out of the cell through diffusion. But for now, small nonpolar molecules like CO2 and oxygen, these can freely diffuse. You need enough surface area, but they're ready to diffuse in and out of the membrane as needed. Sometimes molecules can't freely diffuse through these lipids, but they can diffuse if we allow them to have perhaps a channel or something. So we can have sort of transported, facilitated diffusion like a channel. Here we see an anion exchange protein, it's a channel. We can have transporter proteins or carrier proteins that don't use ATP, but use some other method to allow us to bring things into the cell. By contrast, active transport uses energy in the form of ATP. So we're going to hydrolyze ATP, which in Chapter 9 we'll learn more about why it's so difficult to earn all the ATP that we need, but we're going to be using it here to transport things back and forth across this membrane. And we'll find out that we can actually mix and match some of these categories to transport in and out the molecules that we need. So let's talk a little bit more about what makes a molecule diffusible through a membrane. What kinds of things can simply diffuse through the membrane? So the number one sort of characteristic of a molecule that determines whether it can get through a membrane by diffusion is hydrophobicity. So how hydrophobic is this molecule? So what we're looking at in this graph, on the x-axis, we're looking at what's called a partition coefficient, which is basically a measure of hydrophobicity. Okay, so measuring the hydrophobicity. And on the y-axis, we're measuring membrane permeability. So you can see that basically the more hydrophobic something is, the easier it is to get through a membrane. There are a few outliers in things like water and methanol, but for the most part, these sort of outliers tend to be very small molecules, so this is kind of what helps them escape that. So hydrophobicity plays a large role in whether something can diffuse through the membrane. The second characteristic, which we've already kind of touched on, is size. So we can even think about water versus urea. So water, which we know to be polar, but it's very small, has, just this H2O. Urea, on the other hand, it doesn't seem like it's very large. We're going to have some carbon, some oxygen, a couple of nitrogens, throw some hydrogens on it. Doesn't seem that big, but it's big enough that its permeability ratio relative to water drops significantly. So size is a huge factor, and you can see even at something as small as urea, we're already experiencing difficulties getting out of the cell or into the cell. The third characteristic is charge. So we already know that things that are going to be polar have difficulty getting through the membrane, but if you have a charge, like this hydroxyl group here, this hydroxide ion, big fat negative charge, you might as well forget it. It doesn't matter that this is even smaller than water. If you've got a charge, you're not getting through. So this permeability ratio, you can see it relative to molecular oxygen, it's just really not going to get through. So ions can't get through. And that's going to be really useful for us in a lot of situations where we need to control ion concentration on one side of a membrane versus another. So just to kind of put this in perspective, factors that determine whether a solid can cross a membrane by simple diffusion, again, dependent on its hydrophobicity, how small it is, whether it's polar or not, and if it's charged. So you can see, again, hydrophobicity plays a huge role. If you're small and hydrophobic, into the membrane you go. Okay, oxygen can diffuse in, carbon dioxide out. We've got some nitrogen here. Benzene, you really don't want to diffuse through your skin, but it can. Okay, so this is why we wear gloves when we work with things. Okay, so things that are small, maybe a little bit polar. Okay, urea can get through, but not as much as maybe water or something like oxygen. Okay, so it can't diffuse as quickly. That's what this small arrow is about. Things that are larger, again, glucose, not a huge molecule, but it's large enough that it'll give problems. It's polar. It's just really not going to get through without some help, and ions just turn back now. So keeping that in mind, when we think about the way that we set up carrier proteins in the membrane, channels in the membrane, the way we think about facilitated diffusion. So let's look at some examples of passive and active transport. So in passive transport, the important thing to remember, we're looking at this on the left, is that solids are moving down a gradient. So they should be moving in the direction, in general, of their concentration gradient. So just like with diffusion, we see a higher concentration on the outside. It wants to naturally move towards an area where there's less concentration of this molecule. Passive transport, with facilitated diffusion, we're looking at that same relationship. There's more out here, so it wants to diffuse inside, but we just have to create a space for it to do so safely, so it doesn't have to interact with any of these hydrophobic fatty acids. Here we see a carrier protein. We'll see a couple more examples of these guys. This is a similar idea to a channel, except that it's only open to one side of the membrane or the other, dependent on what's going through it. So again, just providing a safe space for this to move down with its concentration gradient. By contrast, with active transport most of the time, what we're trying to do is move things against its natural concentration gradient. So you can see there are fewer of these molecules out here. Don't want to spoil the surprise, but pretty sure this is going to be a sodium-potassium pump. But you can see that we're dropping these guys off in an area of higher concentration. So we have to use ATP to move with the direction of this carrier, which will be against its concentration gradient. So again, moving things in the opposite way that they would usually want to go, and that's why this requires energy. So let's look at a couple of examples of channels and carrier proteins. So again, a channel is just going to provide kind of a safe tunnel for whatever item it is to go through the cell based on diffusion. So if this molecule is not hydrophobic enough to pass through this membrane, we just simply make a channel that's got properties that will allow it to go through. So let this protein be hydrophobic, and it will block these hydrophobicity issues from our solute, and we can let that guy go into the cytoplasm. Okay, this can also work for things that are going to be charged, like ion channels, or things that are going to be like just too large. So we have also things like carrier proteins, which aren't structured the way that our channels are structured, but they still provide that sort of safe space to transport this protein where it does not, or this molecule, so that it does not have to interact with this hydrophobic plasma membrane on this fatty acid side. So you can see we've got a lot of these molecules out here. Whenever this carrier protein is empty, it's open to our extracellular side. Once one of these molecules binds in the slot, that causes a change that allows it to open to the inside where we can drop off this molecule. So again, these sort of, or just like the name implies, facilitated diffusion. We're moving in the direction with the concentration gradient. We're going along with it. We just need a little bit of help from either a channel protein or a carrier protein. So I want to go into a little bit more depth about carrier proteins. So let's look at some different types of carrier proteins because these can get pretty interesting. So here we see on the left just an example, just like we saw on the previous image. So this is going to be some type of molecule moving with its concentration gradient. You can see that now at this point it's open to the inside, so we can drop that off inside the cell. Okay, so we can call this an example of uniport because one thing is going with its concentration gradient into the cell. Here on the right is where it gets interesting. So we can actually move two molecules at once. And the trick here is that one of the molecules has to be going with its concentration gradient. And that sort of flow can be used to pull a second molecule along with it against its gradient without using ATP. So think about this like a raft being swept along in the river. If you have a river flowing in a certain direction, a raft can go with it, just being caught up in that flow, okay? So that's a kind of a good analogy for what we call symport. So a molecule very highly wanting to go in the direction of its concentration gradient, we can pull another molecule along with it. So we're going to see some examples of coupled transport in just a little bit, but just keep that in mind for now. Unlike our river example, antiport, we can have one thing going with its concentration gradient while the other is going the opposite direction out of the cell against its concentration gradient. So one of the things has to be going with, the other thing can go against, but it can go either the same direction, symport, or the opposite direction, which is antiport. Both of these examples are called coupled transport. So this is a pretty cool example, and we're getting kind of ready to set up for some interesting sort of real-life examples in the cell. So let's take a second, take a break from these carrier proteins, and just for a minute, let's look at some active transport. So we'll come back again to these sort of symport, antiport ideas in a moment. So what is the usual source of energy for active transport? Okay, so we know that it's ATP. That's kind of the hallmark of it being active transport. So in this case, we can see that we're using ATP to drive transport of protons from the inside of the cell to the outside of the cell, and it is against its concentration gradient because we're moving from a less concentrated region to a more concentrated region. So proton pumps are really important. So it's very important to build up these gradients on one side of a membrane versus another. So active transport, again, we're going to need ATP to move something against its gradient. So we've got now our direct active transport. Let's add this example of a proton pump to a real life cell situation where we're going to be kind of adding it to coupled transport in a carrier protein. So let's look at what's going on here. So this is that same proton pump that we just found, okay? And you may have been wondering why we want a bunch of protons out here. Now it's about to become clear. So here we have a carrier protein that is a sucrose proton cotransporter. So this is a coupled transport mechanism that uses symport. So notice the same direction. So in this case, what we really want to do, the whole goal of this is to get sucrose into the cell because sucrose is delicious. We can use this for things, okay? But there's a higher concentration of sucrose in the cell than outside. So sucrose would be moving against its gradient. Here, what we've done is we've set up a proton gradient. We've pumped these protons against the direction they normally want to go, and we've built up a higher concentration. So really these protons, what they want to do is diffuse back in. So that's kind of what they want to do. Okay, so they're going to go with their gradient back in and basically just keep circling around, using energy to pump them out, and they just circle right back in. Okay, so it's kind of like a little current of protons that's just going constantly. And in that current, we're pulling sucrose in with it. So even though sucrose is going against its concentration gradient, it's being pulled in with this proton current. Okay, so technically this is called indirect active transport. Indirect active transport. Okay, but really you can break this down into pieces. The proton pump is definitively active transport. The carrier part of this cotransporter, is definitively facilitated diffusion. So we just have put these two things together and say, okay, sucrose is being driven by indirect active transport. So why is it set up like this? I don't know. Why not just actively transport sucrose into the cell? For whatever reason, this seems to work. Okay, so it may be that this proton pump can power other types of transport mechanisms. So we only have to pump one thing. Okay, it may, just be how things evolve. But the idea again is to get sucrose into the cell. So I wouldn't worry too much about getting caught up in the terms indirect active transport or direct active transport, but more it's just important to understand the relationship between this proton pump and this carrier protein for sucrose and protons, and see how you could potentially set things up like this in the cell. Okay, so now let's look at another example, but this time direct active transport. So I feel like we really can't talk about active transport of ions without mentioning the sodium-potassium pump. The sodium-potassium pump is so important for making sure that cells like nerve cells or neurons can maintain that resting potential. It's really important to maintain that charge in the cell, particularly if we're going to have a lot of ions flowing back and forth as part of a signal. So that's one of the main functions of the sodium-potassium pump. So we'll start out and we'll look at this kind of just step by step, and then we'll see an animation of how this occurs. So here, of course, this is going to be our transport protein. Okay, we're going to again have slots for 3 sodiums. Okay, so again, we're going to go against our concentration gradient. Sodium is low in the cytosol, and it's high in the extracellular space. So we're taking 3 sodiums from the inside of the cell. Okay, so that's step 1. You can see where those 3 sodiums go. Once all 3 have bound to their slots, this is where the ATP is going to come in. So the ATP is actually used, and we're going to become really accustomed to the real way that ATP actually works. In this case, the way that we use it is by transferring a phosphate. So we should be comfortable with the idea of ADP at this point. So we're going to use that extra phosphate and attach it onto this transporter protein. So that causes what we call a conformational change, or a change in the way this is shaped, so that it opens up to the outside of the cell. And that also allows us to kind of squeeze out these sodium ions. So goodbye sodium, we do not want this anymore. Okay, so once it's open to the outside of the cell, this gives us the opportunity to bind potassium. And again, potassium is low on the outside and high on the inside. So we're going to be moving against the concentration gradient. So I bet you can guess what needs to happen for this to open back up to the inside. Make it a little hint down here. So to open back up to the inside, we have to lose this phosphate, which will happen in response to these potassium ions binding. And we open back up to the inside and just finish this up by dropping the sodium off, and it's ready to cycle again. So we've been through the steps slowly. Let's now go ahead and watch our video of the sodium-potassium pump.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Okay, so now we've learned a little bit more about active transport. So the examples of the proton pump and the sodium-potassium pump. And we've looked at some examples of diffusion and facilitated diffusion in the form of carrier proteins and in channels. So again, we'll be looking at channels particularly, and maybe some of these pumps quite a bit more in the future chapters coming up. So it'll be important to kind of get those basics down. But in addition to those types of transport, we have what we would refer to as bulk transport, which can occur across the plasma membrane. So this would be through exocytosis and endocytosis, which we've actually already talked about a little bit. So this may be a little bit of a review. So again, one of the things that we can think about in terms of bulk transport or endocytosis is phagocytosis. So this is like cellular eating. So this is a form of endocytosis, or bringing things into the cell. In this case, this is a food particle. This could be something else. But here we see some pseudopodia sort of extending. So this could be due to maybe some actin going on in here. But we envelop our food and take it into the cell, most likely to merge with the lysosome for further breakdown. This is our food vacuole. And this is really clearly illustrated in this image of an amoeba, which is engulfing a bacterial snack. So this is its food source. And you can see that this bacterium is not that much smaller than the amoeba, but it's going to completely extend these pseudopodia and engulf this bacterium, hopefully to merge it with some digestive enzymes pretty soon. So you can see this guy eating. And this is, to me, is just amazing. It's like one of those images of like an alligator that's being eaten by some kind of really large snake in Florida, and you don't know who's going to end up winning this fight. But in this case, we know that the amoeba can handle it. So We've got phagocytosis, which is cellular eating. So we need to couple that with some cellular drinking, or penocytosis. So penocytosis is similar to phagocytosis, but we're looking at smaller particles. So this is kind of just like an endocytosis, almost sort of randomly. So we're just sort of grabbing whatever's out here in the environment and taking it in the vesicle, and we'll just sort it out later. So this is penocytosis happening in a small blood vessel, and you can see that this is going to be like just all over this cell, so it's happening all along the surface. Okay, so this is, again, we're just grabbing not so much a large food particle, not so much anything in specific, but just whatever happens to be out here in the environment. Okay, so we may take that to the lysosome or elsewhere, depending on what's in it. Okay, so these two mechanisms, the phagocytosis, were definitively just cellular eating. Penocytosis is a little bit random, but we do have a good ability to go ahead and select what we want to bring into the cell, at least to a degree. So we can actually do this with receptors. So we've seen a lot of receptors for molecules as we travel through the endomembrane system. So we can actually have receptors on the outside of the cell, which can trigger endocytosis. So actually things like virus binding to receptors on the outside of the cell can trigger taking the virus in through endocytosis. So this is something that you often see, this sort of receptor binding event allowing viral entry into the cell. So here, in this case, we're not dealing with viruses necessarily. We may be dealing with, whatever other molecules we need. So we can have a lot of receptors, and once we've filled up a group of these receptors, this will trigger our coat proteins. So we don't have to memorize things about coat proteins now, but if you remember back to the sort of KDEL example, we looked at COP1 and COP2 coats, and other types of coats exist, but we didn't talk about them. Again, if you really are interested in this sort of coat protein type of thing, we talk a lot about this in advanced cell. So we've got receptors that are bound, and this stimulates the creation of a coat, which will help make this form into a vesicle, so that we've selected what's in the vesicle. So it's like a very highly selective process, but you can see there's a little bit of randomness too. We got a couple of other things in there as well. So we can look at the microscopy for this. You can see this is going to be our sort of pit that's going to be endocytosing, and we can have receptors that will grab whatever molecules we need out here, and you can see the actual coat proteins forming on the outside. And this is just really an excellent picture. So you can see that whatever this is, we are just really concentrating it inside this vesicle, and you can see beautiful coat protein on the outside. So this is just a really intense image. So again, this is the one way that we can select things but bring it in kind of in bulk. So there's some really cool ways that the cell can handle this issue of getting things across the membrane. So just to conclude, membrane transport generally can be divided on a small scale into simple diffusion, passive transport, which will include carrier proteins and channels of facilitated diffusion, and then active transport. So this is going to be when we use ATP, and we're moving against definitively the concentration gradient. And whether a molecule requires help getting through the membrane depends on its biochemical properties. Is it hydrophobic? Is it small? If you're going to have a charge, you're just not going to get through. So this will depend on its biochemical properties. The cell can also regulate bulk transport using endocytosis and exocytosis, specifically phagocytosis, penocytosis, and receptor-mediated endocytosis. So we're able to control what we get in the cell.